Today, FDA approval sought in urothelial cancer and multiple myeloma, orphan drug status granted in small cell lung cancer, encouraging findings in an ovarian cancer trial, and a Canadian approval in breast cancer. Welcome to Enclave News Network. I'm Gina Columbus. A biologics license application has been submitted to the FDA for enfortumab vedotin for the treatment of patients with locally advanced or metastatic urothelial cancer who have previously received a PD-1 or PD-L1 inhibitor and who have received a platinum-containing chemotherapy in the neoadjuvant or adjuvant locally advanced or metastatic setting. The application is based on findings from a cohort of patients enrolled on the single-arm Phase 2 EV-201 trial in which the overall response rate was 44% with enfortumab vedotin in patients with locally advanced or metastatic urothelial cancer. This included a 12% complete response rate. Additional results showed that the median overall survival was 11.7 months, the median progression-free survival was 5.8 months, and the median duration of response was 7.6 months. Cohort 2 of the EV-201 trial continues to enroll patients. Previously, the FDA granted the agent a breakthrough therapy designation in March 2018 for patients with locally advanced or metastatic urothelial cancer who experienced disease progression during or following checkpoint inhibitor therapy. In multiple myeloma, a supplemental biologics license application has been submitted to the FDA for a new subcutaneous formulation of daratumumab for patients with the disease. The sBLA is based on findings from the Phase 3 Columbus study, which showed non-inferiority in terms of efficacy versus the intravenous formulation of the CD38-targeted monoclonal antibody. Data also demonstrated that the subcutaneous version led to a reduction in treatment burden when compared with the standard version. The application also includes results from an ongoing Phase 2 trial. In Columbus, results showed that the objective response rate was 41.1% with subcutaneous daratumumab versus 37.1% with the intravenous formulation, which met the criteria for non-inferiority. Additionally, the median duration per infusion dropped from more than 3 hours per infusion in the IV arm to 5 minutes with the subcutaneous version. At a median follow-up of 7.46 months, the median progression-free survival was 6.1 months with the IV daratumumab versus 5.6 months with the subcutaneous formulation. The 6-month overall survival rate was 83.0% versus 87.5% for the IV and subcutaneous formulations, respectively. The FDA has granted an orphan drug designation to durvalumab for the treatment of patients with small cell lung cancer. The designation follows the announcement of the Phase 3 Caspian trial data in which the combination of durvalumab and standard etoposide and platinum-based chemotherapy showed a statistically significant and clinically meaningful improvement in overall survival compared with chemotherapy alone for patients with extensive stage SCLC. Results of the study will be presented at an upcoming medical meeting. AstraZeneca, the manufacturer of the PD-L1 inhibitor, previously stated that the safety and tolerability findings of the immunotherapy and chemotherapy combination were consistent with the safety profile of each agent alone. The PD-L1 inhibitor is currently being evaluated in the Phase 3 Adriatic trial, which is exploring durvalumab following concurrent chemoradiation therapy in patients with limited-stage SCLC. In ovarian cancer, niraparib as a maintenance treatment demonstrated a statistically significant improvement in progression-free survival compared with placebo in patients following first-line platinum-based chemotherapy, regardless of biomarker status, meeting the primary endpoint of the Phase 3 PRIMA trial. In the Phase 3 randomized, double-blind, placebo-controlled study, investigators compared maintenance niraparib with placebo in approximately 620 patients with stage 3 or 4 ovarian cancer. Those who had responded to first-line platinum-based chemotherapy were randomized 2-to-1 to receive either niraparib at a starting dose of 200 milligrams daily in those with baseline weight more than 77 kilograms or a platelet count less than 150 K per ml and 300 milligrams in all other patients, or placebo. The safety and tolerability profile of the PARP inhibitor was also found to be consistent with prior clinical studies. Full findings of the PRIMA trial will be presented at an upcoming medical meeting. The FDA approved niraparib for the maintenance treatment of adult patients with recurrent epithelial ovarian, fallopian tube, or primary peritoneal cancer who are in complete or partial response to platinum-based chemotherapy in March 2017. Health Canada has approved neratinib for the extended adjuvant treatment for patients with early-stage hormone receptor-positive, HER2 overexpressed or amplified breast cancer within 1 year after completing trastuzumab-based adjuvant therapy. The approval is based on findings from the Phase 3 Extended trial, in which the 2-year invasive disease-free survival rate was 95.3% in those who received neratinib, compared with 90.8% in the placebo arm for this patient population. At the 2-year follow-up, additional results showed that treatment with neratinib led to a 51% reduction in the risk of invasive disease recurrence or death at 2 years compared with placebo. Updated findings showed that at a median follow-up of 5.2 years, the IDFS rate with neratinib was 90.2% compared with 87.7% with placebo, leading to a 27% reduction in the risk of invasive disease recurrence or death. Moreover, the longer-term data showed that the restricted mean IDFS was 56.5 months with neratinib versus 55.2 months with placebo. The FDA approved neratinib in July 2017 for the extended adjuvant treatment of patients with early-stage HER2-positive breast cancer following post-operative trastuzumab. This week, we sat down with Dr. John Gosney of the Royal Liverpool and Broadgreen University Hospital NHS Trust to discuss the changing landscape in molecular testing.
It's very important that the central reference laboratory and the central unit where lung cancer is managed in the context of a multidisciplinary team or a tumour board communicates very well with the community hospitals. It is still the case, certainly in the UK, that the majority of lung cancer is diagnosed in district general hospitals, community hospitals, and the biopsy, therefore, will be obtained by clinicians in those community hospitals, and the initial pathological analysis will be by pathologists in those community hospitals. And it's very important that those practitioners need to feel part of the entire enterprise, and education of those clinicians is very important, because it's very easy to forget when you're working in a specialist centre that when you get outside into the wider world, there is not that intensity of experience and that level of knowledge. And it's important to maintain those links, because ultimately it's engagement of these very important practitioners at the start of the process which determines the quality of the specimen, the way it's handled by the pathologist, and ultimately determines what we in the centre can do in terms of making the very best of that in profiling the tumour so that the patient can be managed appropriately. So education is very important. One of the major changes which is about to occur in England is the centralisation of genomic testing of all solid tumours into 7 genomics laboratory hubs, GLHs. And this is driven by NHS England and is a very ambitious and laudable plan, because of course it will bring economies of scale, it will bring uniform technology, it will bring uniformity of standards and quality assurance, and there are clearly advantages. However, it will inevitably dismantle some of the very well established and very efficient, more local networks that have been developed over the last decade since we first started profiling non-small cell lung cancer, and which actually work very efficiently. They might not use the latest NGS technology, they might use single gene testing like we do in Liverpool, but they are very efficient, they are very quick, and they're very flexible. And I'm concerned that when this change takes place, at least for a while, there will be quite severe disruption to the smooth running of these local networks. Now, of course, that sort of local organisation is not present everywhere across England. It's very patchy, and centralisation in some areas will bring enormous advantages where there have been problems over the last 10 years because there's not been an integrated system for testing. But we have to be careful that we don't throw the baby out with the bathwater here, if I can put it that way. And there are other problems. For example, NGS technology tends to require more tissue than single gene testing. And we all are aware that the specimens that we often get to test as pathologists are very small, and the advances in the techniques used to get them, like EBUS, radial ultrasound, bronchoscopy, and EBUS aspiration of lymph nodes, which have developed over the last decade, tend to get smaller specimens. And so therefore we have to try and manage that. And inevitably, because of centralisation and large panel testing, the turnaround time will increase. It will be less efficient. So there are disadvantages to this. And currently, this change, this transition, is projected to take place on the 1st of April 2020. So this is imminent, and I'm not entirely sure there's a general awareness about what's happening here. So it's a laudable plan, it's extremely ambitious, but I think we have to be aware that for a while at least this might cause some quite significant disruption with the well-organised, more local testing that's established in large parts of England.
That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.